What's up, 3-4? So sorry, I'm on course today, so I'll be teaching you through YouTube. Alright, hope everyone's alive and awake. How are you doing today? Okay, that's the intro. So today we're gonna continue from where we left off. Just a bit of recap, right? We were talking about the temperature's effect on the rate of photosynthesis. So if you recall, we have three different factors that can affect the rate of photosynthesis. Do you know what are these three? That's right. Right, these are the temperature, right, the light intensity, and the carbon dioxide concentration. Okay, so we have covered two of them already, and now we're going to move on to the third one. Oh, but before I do that, just a little recap. So how does temperature affect the rate of photosynthesis? We know that at low temperature, the enzymes are inactive. There's a low rate of photosynthesis. As the temperature increases, the enzyme becomes more active and the rate of photosynthesis increases. At optimum temperature, the enzyme is at its most active, the rate of photosynthesis is highest. And at above optimum temperature, the enzyme starts to denature, so the rate of photosynthesis starts to decrease until it reaches 0. Okay, so let's look at this graph over here. I think there's a bit of a lot of different things. Where's my mouse? Okay, so let's look at the four different colors. We have purple and green. So we see that for purple, as the light intensity increases, right, the rate of photosynthesis starts to increase until a certain point, right, around this area where it doesn't increase anymore. Okay, so what are the factors here? With that 0.3% of CO2 at 20 degrees Celsius. Okay, then let's compare this purple one to the green one. We see that for the green one, it goes slightly higher, right? And this is 0.3% or not 0.03% of carbon dioxide at 30 degrees Celsius. So what we can see is that when you increase the temperature from 20 to 30, the maximum rate of photosynthesis increase slightly. Okay, not really a lot, because there's other factors that can also be limiting it. Okay, for example, the amount of carbon dioxide is a little bit limiting. There's too little of it. So let's look at the blue and the orange line. For the blue line, we see that it starts to increase as light intensity increases up until around this area, right, where it no longer increases. And the blue line is 0.13% of CO2 at 20 degrees Celsius. Okay, so it's definitely higher than the bottom two. The reason is because there's a lot more carbon dioxide that's being supplied to it. And lastly, let's look at the orange one. So we see that for the orange line, there's 0.13% of carbon dioxide at 30 degrees Celsius. Okay, so if you compare this, the thing that changed was the increase in temperature. And there was such a huge jump in the maximum rate of photosynthesis. That is because with all the other factors, right, 0.13, we can say that carbon dioxide is no longer a limiting factor. It doesn't really matter. There's enough carbon dioxide for it to for the plant to use. So that is not that's no longer a limiting factor. And we see that at high light intensity, we don't need to worry about light as a limiting factor also. So what is more important is then the temperature. Okay, as I shared with you last week, we know that some plants require a certain temperature, especially tropical plants that are in countries like Singapore, Malaysia. These tend to grow better in hot environments. Okay, plants that are living in cold countries tend to grow better in slightly colder environment. Okay, so if you can, right, you take out your notebook, right, and then you just sketch this graph, right, and you can label all of this over here. Okay, so you can pause the video if you need to, and then just finish the sketching. Okay, once you're ready, then you can move on. Ready? Let's move on. Okay, so a few things about the enzymes that are involved in photosynthesis. I told you before, this enzyme, the photosynthesis reaction is enzyme dependent. That means that it can stop working if the enzymes become denatured. So at very low temperatures, enzymes are inactive. Remember, at low temperature, they are not denatured, they are inactive. But at extreme high temperatures, alright, anything is higher than optimum temperature, the enzyme becomes denatured, so the photosynthesis is unable to happen. Okay, so now let's talk about the last factor that can affect the rate of photosynthesis, right? And I spoke a little bit about it just now, which is our carbon dioxide concentration. Okay, so please take out that paper that I gave to you previously, and you can sketch a few things that I say on it. Okay, so here we have a very similar setup to what we saw in the video last time. Right, we got the lamp, we got the water plant, and the thing that we're going to change is our sodium hydrogen carbonate. Okay, the reason is because this substance provides the amount of carbon dioxide in the solution surrounding the water plant. Okay, what we should keep constant is the temperature, and we should keep the light intensity, the distance, equal. Okay, so we use different concentrations of sodium hydrogen carbonate. You can say, for example, 0.1%, 0.2%, so on and so forth. Remember, we need 5 different key points, right? So 0.1, 0.2, 0.3, 0.4, 0.5, at least 5. Okay. So what we see is that when the rate of bubbling increases, then you see the oxygen bubble is likely because the concentration of sodium hydrogen carbonate has been increased. Let's look at this graph. So we have seen this kind of graphs a few times. We know that as you are increasing the carbon dioxide concentration, right, the rate of photosynthesis increases up until the point of Y, where carbon dioxide is no longer the limiting factor, right? Because there's more than enough carbon dioxide for the plant to use. And so the plant then can reach its maximum rate of photosynthesis. Okay, so like what the picture is saying, beyond point Y, the rate of photosynthesis remains the same even though the carbon dioxide concentration is increased. Okay, so if you were to compare these two lines, the green line is 0.03 and the red line is 0.13. So we see that by increasing the amount of carbon dioxide, the maximum rate of photosynthesis increases drastically. Oops. Okay, so you might want to sketch this down on your notes. Okay, so one thing to take note is that when you are trying to do an experiment for each of these different factors, you must keep the other two factors constant, right? You cannot change two factors at the same time. If not, it gets very confusing. So you wouldn't know if the graph is increasing because of a certain factor or the other factor. Okay, done copying? Let's move on. Now we're going to talk a little bit about what happens to the glucose that is made in the leaves after photosynthesis, and following that we will talk a bit about the leaf structure. Okay, so we know that when photosynthesis happens, right, water and carbon dioxide in the presence of light energy and chlorophyll will be converted into glucose and oxygen, right? That is the word equation for photosynthesis. But what happens to the glucose? Now the plant is pretty smart, right? The plant is not going to just, let the glucose be glucose. They're actually going to use it for different purposes. So this is something important. I need everyone to copy this. First of all, the glucose can be used by the plant cell for cellular respiration. Okay, so what is cellular respiration? It is basically the cell's living processes, right? So the cell needs to breathe, the cell needs to do activities, right? It needs to perform certain functions. All of this is classified under respiration. Any of the cell activities require energy, and energy is provided through the process of respiration. Second, the glucose can be converted into cellulose, right? And the cellulose is found in the cell wall. So the cell wall provides a structure for the cell and it protects the cell. Number 2, excess glucose can be stored temporarily as starch in the leaves. So if you recall, all the different experiments that we've been seeing so far have been testing starch, right? For example, you talk about the de-starching experiment, or you talk about the you put a black color paper experiment on the leaf. All of that is using the property of starch with iodine, reacting with iodine to turn blue-black. Okay, so this is quite obvious, right? Glucose can be stored as starch in the leaves, right? You've seen this quite a few times. Number 3, glucose can be converted into sucrose, which is transported to the storage organs. So what is sucrose? Right, sucrose is another sugar, right? It's quite similar to glucose. It's just a slightly different form of it, but the chemicals are exactly the same, right? Carbon, hydrogen, oxygen. It also makes up sucrose and it also makes up glucose. So you just need to change it slightly. And they transport it to the storage organs, which is the root. Okay, it could be the root and it could be certain parts of the plant, maybe the fruits. So let's take for example an apple plant, right? So when you bite into the apple fruit, why is it that the apple fruit tastes quite sweet, right? It's basically because of the sucrose that can be found in the apple fruit. So you can consider that the fruit is a storage organ, or in other plants, like for example a carrot, right? The carrot itself, the root of the carrot is the storage organ, where the sugars that were made in the leaves are stored as sucrose and starch are found inside the root, right? So the carrot, the orange color part, has some sucrose, that's what makes it sweet, and also has some starch. That's what gives it that certain taste. Okay, number 4. Sorry, excuse me. Number 4 is that glucose can react with the nitrate and mineral salts that come from the soil, and they can combine to form amino acids. So if you recall, what do amino acids do? Amino acids can combine to form proteins. Okay, so what is this protein used for? There's a few things. Firstly, it can be used for the synthesis of new protoplasm or new cells. So when the plant multiplies, right? Not say multiplies. When the plant grows, it has new leaves, new stem, new roots. These new cells has to have new protoplasm, and this comes from the amino acids that are turned into proteins. Furthermore, excess amino acids can be transported away for the synthesis of new why is it repeating itself? Okay, for the storage as proteins. Okay, so oftentimes people think of plants, right? They say, oh, plants don't have proteins. But have you heard about people who are vegetarian or vegan people? How do they get the proteins? It's basically by eating certain type of plants that are very capable of storing or converting their glucose into proteins. One example is soya bean, right? Soya bean. So when they eat the soya bean or even peanuts, right, all this kind of thing, they actually contain protein in the nut itself or in the bean itself. Okay, and fifth, the glucose can be used to form fats, and the fats can be stored. The fats can be used for respiration in a certain way, in another alternative way which we won't cover, and also the fats can be used for the synthesis of new cells, right? Specifically the cell membrane. Okay, so I hope you have managed to copy all of this 1, 2, 3, and 4 and 5. Please pause the video at the right timing so that you can copy it. Okay, ready to move on? Let's go. Okay, so this is just a quick overview of why photosynthesis is important. Sometimes people tend to skip over this, but it has come out in O levels before, so I would recommend taking notes for this. Okay, so why is photosynthesis important? It's because plants are the producers in a food chain. So the chemical energy, right, the glucose that can be converted into other things like proteins or fats or sucrose or starch, this is known as chemical energy, right? It can be, all this chemical energy can be transferred to other organisms through feeding, right, through ingestion. So when we humans, when we eat a plant, right, a potato for example, we are basically stealing the chemical energy that was stored inside that potato, and we are adding it to our own body, and that's how we survive. We use that chemical energy to do our own respiration in our body. Okay, so that's the first important of photosynthesis. Secondly, photosynthesis helps to purify the air by removing carbon dioxide from the atmosphere and releasing oxygen into the atmosphere. Okay, so this is good because we know that carbon dioxide is a greenhouse gas, which means that it can cause the earth to become a little bit hotter, right? Not a little bit, actually pretty hot. So in a sense, sorry, purify means that it's not really a purify like a filter but it removes carbon dioxide and it releases oxygen. And who needs oxygen? Of course, is the us, right? Mammals, humans, other animals, even plants, right? At night, plants also need oxygen at night when they are not doing photosynthesis. They need to carry out respiration, and respiration always requires oxygen. Okay, dead plants, right? Especially since ancient time, right? When the plants die, they will be buried underground, and over many years, thousands of years, they eventually turn into coal. So coal can then be burnt and be used as a fuel source. Okay, so in the past, Singapore used to rely on coal in order to generate electricity. So there was a power station called St. James Power Station, and they will basically burn coal. That coal would spin a generator. The generator would turn the coal into electricity, right? In a simplified way of speaking. Okay, so let's see, how long has this video been? Okay, so this video has been quite long already. Maybe what we can do is we'll continue with the leaf structure the next lesson. Wait, so maybe on Friday or next week. So now what you can do is you take the time to pause and analyze certain parts of the video that you didn't catch. Okay, you can do this as a class, or you can do this on your own PLD or phone. And after that, right, please look through your TYS and attempt all the questions that are feasibly done, that could be done with what you know. If there are certain questions that you do not understand, you feel like you have no clue what the question is asking about, then you will need to do a little bit of reading. Most of the things that are coming ahead, the leaf structure and the function, are not too difficult to understand. So if you read your textbook, I believe that all of you can actually grasp the content. Maybe just the way of writing might be a little bit off, but let's try first. Okay, so finish your TYS as much as you can, and let's try and submit it to my pigeonhole, right? By the end of the lesson. Okay, so I think one of you could always help me put the books into my pigeonhole, and when I'm back on Friday, then I will try and quickly mark through and give it back to you. Can? Okay, all the best and have fun in school. Bye, miss y'all.